All right. So first of all, just who am I? I actually have a lot of hats at UBC. I'm involved in a lot of different aspects. So in terms of faculty of science, my day job, what I primarily do is I'm professor in the Department of Chemistry. And my expertise is really synthetic organic chemistry. So actually a fair number of people in the room actually took chemistry courses or are currently taking chemistry courses from me. This is really what I do. We're a molecule building group. That's what we do. Small molecules is really my specialty. Custom small molecules, pretty much anything. If you need to make anything, we can pretty much make it as long as it's a small molecule. The other thing I do is I'm very heavily involved in the safety aspects within UBC. So for example, I'm co-chair of the Faculty of Science, JOSH, which is Joint Occupational Health and Safety Committee. The reason it's going to be relevant for the talk today is this actually has another interesting network of connections that, again, part of the reason I've bumbled my way through a lot of these things. For example, I have good ties with HAZMAT, Vancouver HAZMAT. We have actually a lot of good connections. Unfortunately, with chemistry and the safety aspects, we need to develop good HAZMAT connections, as you can imagine. And additionally, I actually have roles within the University of British Columbia higher up as well. I'm the chair of the UBC Chemical Safety Committee. This is the comparable aspect of the UBC Biosafety Committee, the Radiation Safety Committee. It's a relatively new committee at UBC. And as you can probably imagine, there's not a lot of good regulation of chemical use on campus. So we're trying to actually rein this in a little bit and have a better safety network. Now, the other thing I actually recently am also involved with is Vancouver General Hospital. I am the scientific director for Concurrent Disorders Fellowship at VGH, and you're going to see how I became part of this, involved in this aspect as well. All right, so I think more importantly, especially for today's talk, who are my collaborators? Without the collaboration, none of the projects that I'm talking about today would have occurred. So each of the little stories I'm going to talk about involves each one of these collaborations. It actually starts with Anthony Phillips, who's on the phone call today, or on the Zoom call, if I'm not mistaken. And again, the talk, that's where I'm going to lead off. That's where I'm going to start the collaboration. It's actually my oldest collaboration, the pain addiction service side of my research program. And I'm going to say how I got into it and where we proceeded from there. Then I'm going to go into a story, actually on the pharmaceutical science collaboration side, Professor Sridhar Lee, Department of Pharmaceutical Science. He really specializes in drug delivery. And also with Professor Brett Page, also in the audience here today. He specializes in medicinal chemistry. And finally, the last talk, and probably the one I'm going to talk the most about today, is a collaboration I've been having about 4 years now, primarily with Professor Djavad, University of British Columbia. He's an electrochemist. We're going to talk about sort of the detection side of small molecules. And also with Dr. Julia Azar, he's the head of complex pain and addiction services at VGH. And if it wasn't for any one of these people, none of these stories I'm talking about today would have happened. These were fundamental to everything we're discussing. So I know everyone here is very familiar with the opioid crisis. I don't want to belabor the point. There are a couple of sort of things I want to bring up just because it's going to be relevant during the talk. One is I just like to dispel the classic myth. It's not just the downtown east side issue. The opioid scourge is cross-border. So basically, every socioeconomic group, it doesn't matter where you are. It doesn't matter which region of the province, what region of Canada you're in. This basically affects pretty much everyone. And it also doesn't actually matter the age. The overdose in BC is pretty much linear with respect to age, which is kind of scary. In fact, I just talked about doing a lot of collaborations with Azar. He works directly with a lot of opioid abuse issues. And his youngest patient who's passed away has been under 18. It's been a minor. His oldest patient's passed away is also in their 70s as well. This is, it basically doesn't matter socioeconomic group, doesn't matter the age, it basically affects everyone. So what's the federal government doing about this? What are local governments doing about this? I would think this is kind of interesting. This is something I just honestly pulled off the website on the federal government, what they're doing alone. And one thing I want to highlight here is they committed over $800 million since 2017, just on a subset of issues dealing with the opioid epidemic. And this is the federal government alone. This doesn't include any of the provincial funding on this as well. Now, a lot of this is with treatment, but we're also looking at sort of other expanding opioid agonist therapies, type treatments as well. It's significant. Since 2017, what's been happening, unfortunately, is it continues to get worse. So this graph, this basically shows the number of deaths for fentanyl overuse in BC. This is taken early in 2022. By the end of 22, it continued to spike up. 2023 is also worse. More people died in British Columbia from the opioid overuse than COVID. Right? Actually, more people have died from opioids than the HIV epidemic in British Columbia as well. This is obviously a major scourge and something we need to deal with. It's also not just limited to the lower mainland. That's the other thing I want to focus on, is this is across the entire province. So one thing to keep in mind when we're looking at solutions to these kind of problems is whatever we develop can't just be applicable to the lower mainland. It has to be to more remote regions as well of British Columbia, which is a significant challenge, especially near the high-tech side that I'm going to be looking at. This was a formidable challenge and still remains. And it's kind of sobering. This is actually even an old statistic. This is from 2022. 6 people die per day from drug overuse in British Columbia alone. Right? And then this is even higher today. So what can I do to help? This is really where I was passionate about. There are a couple of things which are not even directly related to my research. In fact, my research program has nothing to do with pretty much what I'm talking about today. It's just something I've been passionate about, something I've been wanting to work towards. So what can I do to help really relies on, well, what do I do? I'm highly trained scientist, but what am I trained in? What can I do to apply that training to this particular issue? So what do I do? Basically pharmaceutical synthesis. That's really my training. The people from my group, if they're not going to academia, further academic studies, they go into the Canadian pharma industry. In fact, that's the vast majority. In terms of my training, most of my friends from grad school they're at Pfizer, they're at the major companies looking at pharmaceutical synthesis. Now, specifically in my group, and this is, I'm not going to go into the details, just kind of what's vaguely relevant for today. We do a lot of single electron processes in my group, historically. So for those of you who are taking my chemistry class right now, most of what we've taught or taken it historically in my chemistry class, most of what we've taken, what we do in that is 2 electron processes, substitution reactions, in the case of substitution. We focus more on the radical processes, single electron processes, and that's part of my specialty. The other thing my group does is we look at fluorine incorporation in small molecules, especially as it relates to pharmaceuticals. Fluorine incorporation helps the lifespan of the pharmaceuticals, makes them more resistant to degradation by standard metabolism, keeps them going on longer. But the other thing we could do, which we haven't done yet, but if you're interested in collaborating, I love collaborating in every aspect. We haven't gone this direction, but we do have the expertise for PET imaging agents as well. So because it's all fluorine, most of these, let's say half of the current PET imaging agents are all F-18 incorporation. F-18 incorporation in small molecules is a challenge, and they're kind of limited in what you can do right now. So we're looking at new methods, faster methods to get F-18 into the molecules to make more effective drugs. Okay, so let's go to the first vignette. The first vignette, again, would not have happened without Professor Anthony Phillips, and we're basically looking at this class of molecules. So the class of molecules, they're tetrahydroprotoberberins. They're sort of tetracyclic structures that's here, kind of concatenated 4 rings. The only difference between most of the tetrahydroberberins is really the oxygenation network on the 2 areas. Sometimes it's methyl, sometimes it's hydrogen, or both, depending on what you're looking at. Now, in terms of, they're kind of interesting. They target both dopamine D1 and D2 receptors. I'm going to let Professor Phillips talk about his research on these in depth. I'm not going to go into exactly what these target and all the interesting studies. I'm going to go for more of the chemistry side. One interesting thing is, just as a chemist, you say they're dopamine targets. I'd say anyone with chemistry background could see that's definitely reasonable, because if you look what dopamine looks like, such as here, you can see that all these structures are basically dimers of dopamine. All right, you can see the kind of dopamine linkage within there. You can see the second dopamine linkage, just without the other nitrogen in there, and you get the same kind of connectivity. So yeah, it's not surprising these are dopamine agonists. Now, where did I get involved? This is, again, one of the aspects of serendipity, which is what often happens in my lab, is a company will contact me. In this particular case, it was Panora Pharmaceuticals. They are since no longer, I think they've gone out of business since, but at the time, they look for contract synthesis. So contract synthesis is extraordinarily expensive if you actually go to companies. So just for random trivia, the cost of one full-time employee synthesis person on any given project is about $35,000 per month. Right? So most syntheses are multiple months. You're talking about hundreds of thousands just to make simple molecules. Right? So the one that they wanted me to make was one of these HTVs, the cephalidine. This one is basically structured here. You can see same concatenated network. You got the methyl ether there, hydroxyl, methyl ether, hydroxyl here as well. All right. So we quoted about $25,000 for 6 months worth of work because there were no what's called an asymmetric synthesis. We had to set this particular stereocenter to get the proper configuration. Now, at the time, Panora Pharmaceuticals thought that was kind of expensive for only a couple of hundreds of milligrams of things. So they actually hired an independent chemist, and that chemist, they rented out a lab space, and they spent a year trying to make the thing. And this is point 2 of serendipity, is they came back with some really interesting and weird results. They couldn't kind of figure out what was going on. So Panora contacted me again to try to help them suss out the mixtures of what they were doing, what they made. And the interesting thing is they didn't actually make, they had some cephalidine in there, But there's also a lot of the mixtures were contaminated with what we found out was we identified as govidine. That's one of the things my lab can do is look at structures, figure out the overall connectivity. As you can see, staphoidine and govidine have a very similar structure. The difference is the position of methoxygroup. It's just a Mason versus floating. Right? Now, what's interesting about govidine, in fact, Professor Phillips and Honora are very interested in this. At the time, there weren't many studies on this particular molecule. The ICP space was wide open. It's difficult to obtain through natural isolation, which is also kind of an interesting aspect, which is why it hasn't been studied as much. And there are no enantioselective synthetic routes described in the literature. So from my perspective, quite interesting as well. All right, so then we established a collaboration with Honora Pharmaceuticals. We got NSERC funding to be able to do this because we got to do the interesting side of things again, and we went through it. So I'm just going to generically say, when I say planning a synthesis, I'm just going to talk about what I'm talking about, and then I'm going to kind of break it down. And then for the synthesis people in here, I will, for the chemistry people, I'll go a little bit more detail, but not too much. So when I talk about planning a synthesis, there are like two different levels I'm referring to. The first is medicinal chemistry approach, which is basically you just want to make the molecule by any means necessary to test it out. So when a pharmaceutical company hires medicinal chemists, it's basically something myself or Professor Page, and you make a whole library of molecules. You don't care about yield, you just care about purity. You just need milligram amounts to actually test these things. You don't need very much. The next step, of course, is the process chemistry approach. Right, which is once you have a hit, you go to process and then you scale up from milligram quantities to actually be able to synthesize these on multi-ton scale. Right? It's a very different approach often. Sometimes you can use the med-chem approach, but sometimes you have to expand it out. When we plan syntheses, we like to kind of split the difference. We can't go directly to a process chemistry approach. It's impractical. It's almost impossible to go directly. We like to see a med-chem approach that wouldn't take much to be able to kick into a process, some minor optimization. So just as a fun quote I love, this is from Sir John Cornforth. He's describing process chemistry or process chemists love. He says, it does, for example, no good to offer an elegant, difficult, and expensive process to an industrial manufacturing chemist. Industrial manufacturing chemist is basically a process chemist whose ideal something to be carried out in a disused bathtub by one-armed man who cannot read, the product being collected continuously through the drain hole and 100% purity and yield. Trivial process. That's what they're looking for. We're not there, of course, quite yet for what we need to do, but this is the overall goal. It needs to be cheap. For most of the stuff I do, there's a science element and there is an engineering element to what we do as well. So whenever we're doing the fun med-chem synthetic aspect, we always have to think of the engineering side that eventually these things have to be scaled up. In fact, actually, a lot of what I currently do for other companies is working on scale up for what they do. They have a med-chem process and we kind of help shuttle the process through to the process. So here's the synthetic plan. Again, I'm not going to go into the gruesome details of everything here, but I just want to give generically how you disconnect molecules such as this. And usually what you do is you look at certain motifs within the molecule, and you look to see certain structures, and they kind of make sense in terms of how you disconnect based on reactions you guys have learned before. So for example, within this ring, you can imagine that you could break apart this with an extra carbon. That's not an uncommon reaction. Basically, it's an imine cyclization for those of you who have actually done that kind of chemistry. From there, this actually gives an opportunity to set that critical stereocenter. All right. And what we're basically doing from the cyclic imine, you can do an enantioselective reduction. This is certain technology which you can deliver the hydride from one face or the other. All right. So that's our key third differentiating step. And the advantage for what Professor Phillips wanted and Honora wanted is we can make either L-govidine or D-govidine depending on what particular one they want. So this route can access either. It also can access it from a fairly late stage. So we can get all the way through on the racemic synthesis or achiral synthesis and set the stereocenter last in the delivery. This can just be made through deoxide cyclization from this particular amide. And once you're an amide, that's pretty standard chemistry. You go from the amide coupling steps. All right. Now, when you're developing these kind of routes, the other thing you have to think about, of course, is if every step, let's say, is 80%, you start losing material quite quickly from step to step to step to step. And these steps, like, we thought it was going to be roughly 10 steps, 12 steps. You have to have a ton of this. And you have to make these too. These are also not cheap commercially. So we kind of want to go from as cheap materials as possible. So the nice thing about this is both of these can be actually synthesized from vanilla. All right, vanillin, if you ever have vanilla, you can buy it's expensive if you get the extract. Synthetic vanilla is the same thing. Right, exact same thing. The only difference, of course, is Actually, the natural actually has a lot of other stuff in there. It's like the nose a little bit cleaner. You can either make it from petrochemical industry. They basically formulate, you basically go there and formulate last stage. Or actually these days, kind of random trivia, you can make it from lignin, the wood byproduct. So 15% of the market's actually from a lignin byproduct. So anyway, it's readily available. It's cheap starting material. Now for the chemistry people in the room. I apologize for those others. This is gonna be a little bit of chemistry. Yeah, exactly. Study up. Vanillin, all you have to do is benzylate it. It's basically an SN2 reaction. What we're doing is we're taking this alcohol, what's called protecting it. We just want to take the alcohol, keep us an alcohol roughly through the synthesis. We just want to make sure nothing touches it. Then we're going to reduce the aldehyde to the alcohol. You turn that into an acid, into an alkyl chloride, then you displace with cyanide. All right. And both those precursors, you can access the same material. The nice thing about there is at this point, you can split bullet. One of them you just have to reduce with chlorine. You take the nitrile for the amine. The other side, you can just hydrolyze the nitrile with vaccine hydroxide to get the carboxylic acid. So the other thing you want to do is you want to minimize total step count. So the fact we can actually make both of these from a similar intermediate really saves overall synthesis. Now from there, so keep in mind, this is 4 steps to there. So total step count 6, but 5 in the longest linear sequence. And then all you have to do is make the amide just for fun. I know you would say you can't do this historically in chemistry, but if you actually take these as a melt, you can actually sometimes basically take the 2 solids and heat them up to a melt. You can actually get the coupling. This coupling work. And then we do the cyclization. It's a deoxycyclization. Then we can do what's called a Nairi transfer hydrogenation. Nairi won the Nobel Prize in 2001 for his asymmetric analysis work, and this is one of his nice syntheses. So this is our nice particular technology. We can access either the R or the S, depending on which one you want at this point. And that's just a matter of formulating and cyclizing. We get to this ring, and then if I said protecting group, you protect the benzyl groups at the last stage, hydrogenation, get it through. So overall, we're about 10 steps to synthesis. I've now been working with Professor Phillips. I can't even remember how long. It's probably been at least 6 to 8 years on this particular project. We keep supplying his group. We are the only source of govadine in the world. So we keep synthesizing this for his group. And we've made now gram quantities of both the D and the L-govadine for the studies will be needed. But this is the advantage of collaboration. Professor Phillips has an expert at actually taking this, checking out particular site, the active sites, what you're going through. But if you actually need the material, this is what the chemists are for. All right, we can synthesize anything. In terms of what's next, future directions on this project, one of the critical things we need to do, I think Professor Phillips would agree with me, Ethan is just listening on the call right now, we gotta make it cheaper. Right now, we're making a gram at a go when we go through the synthesis. And it takes about 1.5 months of postdoc time just to get through a gram of the synthetic route. Because every step, there's purifications, it's not a simplistic process. We need to now take this from more of the med chemi route to the process scale. And the challenge there, just for random trivia, is actually purification is the hardest thing. Purification is the thing which is prohibitive at scale, which we need to work on. We need to work on derivatives with improved aqueous solubility. Often govadine right now, or in fact, govadine needs DMSO to help solubilize it up, be able to use it overall. We can actually make derivatives of this to help the aqueous solubility of this thing. And there are a lot of opportunities with that. And then this is another reason why I like to work with pharmaceutical scientists, as Brett Page and other people there, is drug delivery options. Especially if something which is not very soluble, there are a lot of drug delivery opportunities in pharmaceutical science that we can work on to try to address this problem. All right, that's overall the first vignette. The second vignette I'd like to talk about is one with pharmaceutical science. This one is with Jidar Lee and Professor Brett Page. So it actually all started with just a fun conversation with my good buddy, Brett Page. I am on a million committees in pharmaceutical science. Anyone who does any chemistry in pharmaceutical science usually asks me to be on their committee. So one of the reasons I'm busy is I have a million committees. I'm always on department in pharmaceutical science side, which I love actually to do. I always support my pharmacy colleagues. And we were talking about chemistry before one of our particular meetings. I was talking about recent research in the group. So again, what's my real day job is actually do a lot of synthetic technology. So we recently been working with sulfur fluorine gases. Sulfur fluorine gases such as sulfuryl fluoride and thionyl fluoride. For random trivia, sulfuryl fluoride is actually a commodity chemical. It's dirt cheap. The reason it's a quantity name is bicane. It's used as a fumigant. So I grew up in Southern California in warmer climates. I don't know if you've ever seen this before, but it's called tenting a house. What they do when they tent a house, they put a big big, huge, like circus tent over the house. Then they pump it full of Vicane with the sulfurofluoride and it kills termites. Right? So as a chemist, it's kind of interesting, not because because it's cheap, because that cheap side of things on the engineering. But fluorine is also really interesting in terms of its reactivity. And that's what we've been exploiting in my group. I was telling Brent, that was sulfurol, actually thionyl fluoride, something else we've been working on. It's great for peptide coupling. So we can take amino acids such as this, we treat with thionyl fluoride and pyridine. In only 30 minutes, we can make the acyl fluorides. Now, acyl fluorides are actually somewhat difficult to make historically. They're hard to synthesize, hard to make. The nice thing about thionyl fluoride is the byproducts are sulfites. They just wash out with aqueous washes. You don't need chromatography. It's really easy to purify this. Then you get the acyl fluoride intermediates, which are really well known to undergo peptide couplings quite effectively, in fact. All right, so globally, what we have here is a 2.5 hour at most peptide coupling method. In terms of our method, it's competitive with the best in the literature. All right, in terms of the yields you're seeing, dietary selectivities, it basically is as good as anything that you see, especially this example here. This is a natural amino acid, it's phenylglycine. These, on a chemistry perspective, they tend to ephemerate. So you tend to get both stereoisomers during the course of the reaction. We get a little bit of that, but again, most of the other technologies are even worse. So this is actually pretty good. The most important thing about all of this is you don't need purification. You need simple aqueous wash and it's done, which is why this chemistry is powerful. Now, what's exciting about this is this starts to be competitive with one of the classic ways of making peptides with solid phase synthesis. So if those of you who've worked with solid phase or peptide synthesizers, it's all fundamentally based on this gist of technology. The basic idea is you take your amino acid, you hook it up to polystyrene resin. That's polystyrene resin's functionalized because of amino acids to it. You do your coupling, and then for purification, rather than chromatography, you just wash it off the bead. Then you do your heat protection, and then you make the next peptide. You basically do the reaction. You keep growing the peptide chains on the bead, and because you've got a lot of reagents, a lot of coupling reagents, you can get basically force 100% yield at every step, or at least close to 100% yield. So that's how you can get these really long peptides. Solubility is not a problem, it's always attached to the bead. But what really this solves is both length and it also solves purification. But with our technology, it's column-free solution phase. We can do the same thing because it's sulfite salts. We don't have any peptide coupling regions which are difficult to remove. All we do is you can make the acid fluoride, do your coupling, do your wash. That's every step here, it's just washes. Right. And what we can do is we can make tripeptides, we make pentapeptides, tetrapeptides, pentapeptides, good yield. These are all on scale. These are all reasonably scale reactions. Right? The beads have a problem because you can't have very big scales of them. So you see a lot of these drugs out there are quite expensive, like Ozempic has really long polypeptide chains. The reason there's a shortage is it's really hard to scale up from a chemistry perspective too, because it's all based on these synthesizers as well. We don't have that problem. This is all solution phase. We can just scale this thing up. Now, we are limited to about, say, octapeptide in terms of the length we're at right now, just because the solubility of the peptide itself becomes a problem. But even more impressively about this, my student made this in a day. He made multiple 100-gram scale, multi-milligram scale, I think hundreds of milligram scales in a single day, which is, again, we can do this really quickly and really efficiently. And just further what we've been able to do is we worked at different reagents we've developed in my lab. So actually, this was all done by an honors fourth-year undergraduate in my group, Kathleen Down. She was fantastic. She's now at a PhD at MIT. And over one summer, I gave her the basic idea of what I wanted her to do in the lab, and she synthesized like 40 different reagents, of which this methylmidazoleum sulfonyl fluoride, I have to call MISIV just for short, so to keep it a little easier. This thing actually has the same kind of reactivity of thionyl fluoride as the same kind of reactivity of sulfurofluoride. Now, when we look in terms of peptides, all been done by Joey Lai, he's a graduate student from my group. And what he's found recently is you can just take 2 peptides, you add in MISIV, 15 minutes, you get your product. You don't need 2.5 hour couplings. These are 15-minute reactions. Same kind of purification, simplification. It's really easy to do. So that's what I was. Oh, the nice advantage of this is not only do you need to do each of these individual steps where to make the acid fluoride here, you just mix it. Rather than wait 2.5 hours, wait 15 minutes. Then you wash it, do the next in 15 minutes. And this is the kind of technology which is, we're quite excited. In fact, we're actually working with another colleague right now on automating this with robotics to see if we can actually turn this into a good synthesizer that way. Actually, the thing I'd like to do too is 96 well plates, you just gas the entire plate. You don't even have to add the reagent individually. If you gas the entire plate, you should get the entire plate to do peptide couplings. And it's a really good way to do library synthesis. Nice thing about this, again, is to make tetrapeptide or tetrapeptide peptides and so forth even longer. So this is what I'm saying about serendipity. I'm talking to Professor Page about this new technology. I'm kind of excited from a geeky perspective, from the chemistry side. He's like, we have a lot of projects here in pharmaceutical science that involve peptides. And I think he had a project with a peptide project he's working on. And one we were especially interested in was actually something Professor Shidar was working on. So what Professor Shidar was synthesizing was enkephalin. So maybe you may be familiar with this particular molecule. This is the leucine enkephalin. There's another variety with the methionine over there, but this is the one that we're studying. And these are interested in pain regulation. Again, opioid side, where I'm kind of stumbling into this project through serendipity as well, right? Binds the opioid receptors. It's actually quite good on the opioid side in terms of tolerance. The problem is it's a drug. Peptides are usually pretty awful drugs. The problem with these things is rapidly degraded by peptidases. You have to really modify these or play with these to make it better. And also the charge termini. For those, again, in 233, there's going to be charge termini. So you're going to have ammonium, you're going to have the carboxylates, and that's going to prevent this from going to the membrane easily. Even though you have a pretty hydrophobic core, those charges really hurt the membrane permeability. So what Professor Sheelar did before we'd even had this discussion, he'd actually done his group, because pharmaceutical science has synthetic capability. They had made about 10 of these things, and they're looking through the possibilities. And what they did is they end capped the ends. They had some interesting end caps, some interesting opportunities. And I'm just going to highlight one of the interesting things. So KK103 is one of the end cap ones. Rather than go through the details, that's kind of the gist. And it's shown to act through the opioid receptors in the central nervous system. And what's exciting, this is, again, because I've been doing a lot of opioid projects, which is in contrast to morphine, it was longer lasting, did not induce breathing depression, physical dependence, tolerance. So basically, it didn't have any of the awful effects of morphine. It didn't have the potency we needed quite yet, but at least as the first round, this was pretty exciting in terms of molecules. So I was very intrigued about this as a safe and effective analgesic, or even as a peptide opioid agonist therapy used for this. So this is where we started going into a fun, again, leveraging the community. He's an expert in drug delivery. He has some synthesis. I can really help out in the synthesis. That's really what I do. And Professor Page, he's really the med chemist. That's not what I do. Like in terms of taking that next step, that's what he can do. And he also says, we all have enough synthetic background. It kind of helps for the discussions as well. So Joey Lai was looking at the enkephalin synthesis, and we can basically make this, it's only 4 coupling reactions. You have a 50% yield, but we can make this on hundreds of milligram scale. All right, 2 days. The other postdoc was over, I think, a year's worth of work for like 10 or 12 peptides. We made that in 2 days. And now we're looking at derivatizing the end caps and trying to do other things. Now, the problem with the enkephalins, of course, is they're not as potent. Like, it's not going to be a drug, to be honest. No matter what end cap you use, it's not going to be a drug in its current form. But the next steps as part of this collaboration is you look at more potent derivatives. So if you look at DEDPE, like this structure, if you look at the peptide chain, I'm just going to highlight this. It actually has the same sequence as the enkephalins. But it's conformationally restricted. It actually fits these binding pockets more effectively. And this is much, much, much more potent. And then, but again, it has the degradation issues as you'd imagined before. So one thing we're looking at now, of course, is synthesizing this and then doing the end cap modifications to try to use the two together to try to look at more interesting opioids and opioid agonists. Let's make sure I'm doing okay on time here. Oh, fantastic. Okay, final project. This one is actually the one that did not start by me bumbling into it. We actually had a really good goal here. And this is actually probably my longest story I'm going to be discussing today. This story all came through primarily collaboration with Professor Dan Bozzato. But again, it would not be in its state right now without Professor Quinn Azar as well. He's critical for the development of this. And what we're really looking at is detection rather than drugs for this moment, for this particular point. I'm just giving you a little history of opioids, a little bit of fun trivia to kind of mix up a little of the chemistry, and then we'll get into things. We'll just start with heroin. So heroin, of course, is synthesized from morphine using acetic anhydride. For random trivia, I love putting random trivia in talks. When you get drug sniffing dogs at airports, they can't obviously smell heroin itself because heroin is just not volatile enough. So if the dog actually smells acetic acid from acetic anhydride. Right, so there's a little bit of that, especially if it's not purified very well. It's a little bit of acetic acid floating around. So that's what the dogs are actually smelling as the reagent. Anyway, in terms of why it's called heroin, it's actually named from the German word Beruhig, because it's basically Bayer by Bayer, right after they synthesized aspirin. They did all this acetylation chemistry, looked at synthesizing this, and it was so much more active than morphine. They were quite excited, which is why it's named heroic or strong. Interestingly, it's actually marketed originally as a cough suppressant. I don't know if you guys know this, but it's kind of, anyway, bad idea for cough suppressants. Interesting, of course, heroin's been a scourge in and of itself. I don't want to minimize the impact of heroin and how awful heroin is as a pharmaceutical drug. Obviously, fentanyl is much, much worse. So let's first do, from a chemistry perspective, what's kind of going on here. Usually we draw the structures as kind of flattened 2D perspective. We actually did a 3D. This is the picture of morphine. You can kind of see it's kind of a caged-like structure. If you look at fentanyl 3D, you can actually see some structural similarities between the active sites. In fact, they have very similar type of active sites, the targeting between the two. All right, you see the six-membered the pyridines into there, the arines again blocking the back half of the thing. Of course, between the two of them, lethal dose of heroin is under 50 milligrams. Fentanyl's 2. It is really toxic and really deadly. What's even scarier is on the streets. If you look at, this is an interesting study done out of actually UBC, where they looked at the fentanyl concentrations in various samples on the street to see what weight percent they were at. And it ranged from anything from basically hardly anything just over 0 weight percent, all the way up to a quarter weight percent. Right? There is no standardization of a lot of the street samples of it out there. Part of the problem with the opioid epidemic is people don't know what they're even getting or using. Okay. Now, this, of course, the derivatives are even worse. So fentanyl is 2 milligrams. Carfentanil, which is a elephant tranquilizer, is 0.2. Now, from a chemistry perspective, bringing this back, this is a detection challenge. All right, it's an analytical challenge. If you're starting to talk about A, the concentrations, the range, B, the toxicities and the lethal doses, even the low end here becomes toxic and dangerous and deadly. So what's currently out there? Fentanyl test strips. That's probably the cheapest option out there. The nice thing about fentanyl test strips is they're cheap. They're really cheap. They're point of use. Anyone can use them. In fact, they're quite easy to use. They're full of hand-ons. Problems are qualitative. It's a yes-no answer. And the other problem is they are very sensitive. So that yes-no answer doesn't tell you a lot. So talking to hazmat, they think they're useless from a hazmat perspective. Because if they go into, let's say, let's say apartment that's been used for mixing drugs, everything lights up, whether it's going to be deadly or not. Right? And this, unfortunately, the current drug supply, whether fentanyl or not, has so much trace fentanyl around that a lot of stuff lights up. So even as a yes/no answer doesn't really stop people from using. The other option, of course, is spectrometers. Now, these are expensive, limited accessibility, or more limited accessibility, but they are quantitative. Now, whenever you hear about spectrometers at, let's say, parties or I guess the concerts, they often have them set up, or downtown east side, they've got the drug testing. They're all IR technology. The advantage of IR, the reason they use IR in that infrared spectroscopy in that particular application is it's one of the cheaper spectrometers out there. They're pretty easy for powdered samples. It's quite easy to do the test. The problem is its limited detection is about 10 weight percent. So you're actually missing a good chunk of the samples are out there. The other problem with IR is sugar contaminates the sample, or the masks the fentanyl signals in there as well. And often these samples are cut with sugar, right? So again, major challenge. What is the gold standard? Just use an LC-MS. That will work, no question about it, right? LC part separates, you got a mass spec in there as well to get further compound identification. The problem with mass spec or LC-MS, A, it works, it's great, but they're really expensive, they're hard to maintain, and you need expertise to be able to do this. And if you remember that map originally of where the problem is, it works great in the lower mainland, and that's about it. And even then, like I toured the St. Paul's analytic department, so I was curious to see kind of what they had there. They have LC-MSs going constantly, and most of the samples they take or go through the LC-MS. But people working on the opioid epidemic just don't have access to that. And it just takes too long to get the samples back. It's just not really a viable option, too expensive, too hard to maintain. And especially for clinics across British Columbia, just not much. So this is when one of the few times I'm going to say, I actually planned it from the beginning, electrochemical detection. This is one we're very interested in. We're really interested in this because they're cheap. It's the core technology is used in blood glucose monitors. Breathalyzers are electrochemical in basis as well. right? It's dirt cheap technology. You can have point of use care, but you need the expertise to be able to actually develop these things. And what we're looking for is going to be more complicated, unfortunately, than what these things are. So, initial collaboration. As I mentioned before, I have a lot of expertise in single electron processes, which is actually the first step in a lot of electrochemical. It's a lot of single electron movement in that first step. We have a pretty good sense of what's going to happen. I can look at a molecule and have a pretty good sense of where its redox potentials are going to be. That's just the expertise of the group. I am not an expert in detectors. That is not my, I'm not an analytical chemist. That's not my training. So this is why I love to collaborate. I love to break out of my own expertise. And rather than me trying to learn it, which is a waste of time, I can talk to another professor at UBC, Professor Djavad. We've done electrochemical projects in the past, and he's a specialist in analytical electrochemistry. In fact, he knows infinitely more than I do about said subject, which is why I love to collaborate with him. It's funny, we're both in the Department of Chemistry, but we may as well not be, because our backgrounds are so orthogonal. I show a molecule picture to him, like my entire first half of the talk, you go like, I don't understand any of it. And then when I go through his electrochemical side, I'm like, so it's a good collaboration. It's a nice orthogonal background. I'm just going to do a quick summary for those who wonder what electrochemical. I'm going to show a lot of signals. I just want to give you an idea of what the heck I'm talking about. It's actually fairly straightforward. This is like your classic CV. All right. It kind of looks like a duck from those you remember back in your funding. I would say it's a duck with the head, you know. The basic idea of what you do is you sweep your potential. You start at low potential and basically keep trying to pull electrons out. And at some point, you're going to hit the point where you can actually remove an electron or oxidize your subject species. Then you'll see a signal. Then you sweep it on the reverse end. You start putting electrons into the system. Whenever you see electrons pop in, you're going to see another signal. For the purpose of today's talk, we're just going to focus on the oxidation. It won't be quite like the CV. There's another depiction of it, but the gist is the same. We're going to look at the potential. We're looking at what's called a square wave. This is the derivative of this. So don't worry, this concept same. I'm just looking at the oxidative side and just looking in the forward direction. Just as an example, if you look at fentanyl, if you think about which electron's the easiest to remove, you use all your classic second year principles to figure this out. You think, okay, what's the most localized, which is the easiest to remove? Actually, the nitrogen in there is going to be, goes along there, or be the first ones that are going to be oxidized. So what we did, in fact, Dan and I, over the winter holiday about 4 years ago, we didn't have any funding for the project. I didn't want to put grad students on this because I didn't know where it was going and I didn't have the expertise to really be confident I wasn't wasting their time. So we just went in the lab and did this ourselves, just for fun. All right, we ordered up the samples and we decided to see what would happen. So we ordered a whole bunch of, actually, you can order all kinds of interesting analytical standards. So we ordered up heroin, small amounts, not dangerous amounts. And we did, this is the electrochemical signal. As you can see, you get one oxidation peak about 0.8, which means it's somewhat difficult to oxidize. The further this way, the easier, the further that way, more difficult. And that's probably the little pair on the nitrogen right there. We looked at cocaine similarly. You got the preamine there, which actually has a pretty similar peak. Because it's fairly diagnostic peak later. MDMA has two interesting peaks. Actually, an interesting peak. This one's kind of curious. It's about 0.2. Then we've got the latest. This is a little more challenging to discuss, but this one is the immune peak in there as well. When we got the fentanyl, we got really excited because not only do you see the peak around 0.8, you see a peak around 0, just past 0, and it starts to grow over time. In fact, fentanyl actually electrodeposits on the electrode. So even small amounts, you can start concentrating up the electrode, you get more and more signals you do it. So it's actually really good for low concentration analysis. So this is the part we're really focusing on for detection. We're curious again, is this unique to fentanyl? If you have different analytes, what can you do? So we looked at fentanyl, carfentanil, alfentanil, norfentanil. And curve in blue here is fentanyl. You can see slight shift. Well, one more thing, norfentanil, this is not a very active, this is just sort of a control that we're using. And you don't see much of anything for norfentanil. Carfentanil shifted as a double peak, which is kind of interesting. And then the alfentanil is also shifted across. We actually do get a different response for the different fentanyl derivatives, which means that the potential for electrochemical device to be able to analyze each of these derivatives. The basic idea we had then for our detector is if you have all the analytes in solution, we just need a couple different axes of differentiation. So we can get a separation, not as good as an LC separation, but we don't need it. You just need a little bit of pulling in one direction. Then you do your electrochemical analysis, you get a little bit of differentiating the different axes. And then you can do a couple other electrochemical gains. You can keep adding to these axes, and you get enough separation that you can start doing principal component analysis on these things to be able to suss out what you've actually gotten. So for example, we just did over the holiday, we just did a quick study of something containing all of these. And what we're looking at here, each one of these was a separate CV, and each of these is over time. So as the thing's eluting across the separation column, you start pulling out these samples differently. And each one of these we can then distinguish and deconvolute based on what was actually the concentration in the mixture. All right. So what do we need to make this quantitative? One thing we need to do, of course, is we need internal standards, right? Especially if you don't know what you're starting with. Figuring out the weight of some of those samples is really difficult to figure out. We need a standard in there. This is an ideal window for your internal standard. You need to develop something that's kind of away from everything else, okay? So how do we do that? What you need is a small molecule synthesis expert, me, all right? I can help design the molecule to push that redox window into where you need it to be. So we did some initial studies, actually under IP protection. I'm sorry, I actually can't tell you about this particular one, but our initial studies, we've got them into this window. They're not perfect yet, but at least we've got kind of the direction and the range of what we need. So once we did this in lab, we thought, oh, this would be exciting. We started with hazmat because I had the good connections with them. Fire departments across Canada, we pulled very interested in technology. Police less so because they wanted the hazmat to deal with it rather than them dealing with themselves. We went to NSERC because we have an electrochemical detector. It's cheap. The core technology is easy and can detect pretty much every fentanyl derivative out there. They said, not broad enough market. All right. I was like, so we couldn't get funding for it from NSERC. Right. So it was like frustrating. This project was sometimes. So this is where serendipity kicks in. Dan and I are like, okay, what's next? Because we wanted this to move. We needed this to move quickly. We're going to go through the next round of NSERC anyway. It just so happens that my daughter's buddy's mom is a therapist at BGH. All right. And she happened to know Dr. Puya Azar. And I was complaining to my daughter's buddy's mom about the funding situation, about all of this. And she said, oh, I know someone who works on the opioid. You should do it, Dr. Puya. Talk to Dr. Azar. So we had a meeting with it, and he was super excited about the technology because he had a different application for it. And this is going to be a moral life lesson, which is it's better to start with the clinicians first in the future rather than try to find the application early. And we'll talk about that a little bit later. And his idea is basically he's looking for opioid agonist, or basically treatment. So if you have opioid agonist therapy, the thing is if you go in the hospital, they start you on low doses because they don't want to overdose you. And they slowly titrate that amount up as you go. So in extreme cases, they'll start higher, but for the most part, they start low. And the standard of care is about 90 days to get effective dosage by this titration method. And the problem with this is most people relapse before you can get there. Right then, for that, Dr. Azar is working on other options to deal with this as well, but this was the problem at the time and still is a pretty significant problem. So he's excited about this. Well, if I knew what their opioid levels in the body were when I started, I can start higher. And then we can actually get to the target dose a lot faster. The problem with that is, of course, you need an LCMS, which are really expensive, not available in clinics. So that's why he wants a clinical opioid analyzer. Because if you had something, if you could take saliva, blood, urine, anything from a patient, you know what the opioid levels are when you enter a clinic. You can then immediately start them on a higher dose. And you can be sure that you're not going to overdose them at that particular point. At least get them off the fentanyl fast. So at this point, we assembled a team, and we actually got funding for this. We won the Dr. Daniel Jung. He's a postdoc who participated with Dan with auto health development. So we actually started being. Oops, the next connection is unstable. I apologize to those on Zoom. All right. So where are we at on this particular project? Now that we have money, after we have a lot of money now flowing into this project, the first thing we need to do is to test his idea in the first place to see how viable this is. So we actually are now, we just got ethics approval for a clinical study to actually look at opioid levels and to see how well it matches in terms of blood, urine, saliva, see which of these is the best one to be looking at. Next issue, of course, is detection limits. Everything we've done at this point was really focused on, like drug detection and like powder samples. If you go to physiological it's a lot lower concentrations, and it's a lot bigger challenge. So the first thing is we need to see how low detection limits we can get. So this is something I've been working on with Dan Bozzato. What Professor Bozzato is doing is he basically takes this electrode and he starts to electrodeposit graphene oxide. You basically increase the surface area of your electrode, and you can get much better detection limits. And that's what they're looking at with this. And what my contribution to this is small molecule synthesis. We need to do a couple test compounds as we're making these molecules. So in terms of detection limits, basically this shows kind of how low far down on the detection you can get. This is the bit overall, but the take-home message is we could get less than 10 part per billion using this particular technique, all right, which is well within physiological fluids. In fact, for addicts, it's probably getting higher than that. This is one of the reasons we want to do that clinical studies. I don't know what our target is, but 10 part per billion is well within. I think if you go to a hospital and they give you fentanyl, I think you're around 10 part per billion at that point. So I'm pretty sure it's gonna be higher than this, but we needed the study. We can also, again, this surface modification, we got better absorbance properties. The issue, of course, is biological fluids for electrochemical make it more complicated. So there's a lot of proteins in the blood that mess up electrochemical detection. There's a lot of things in urine. There's a lot of things. Saliva is actually one of the cleanest, ironically, for electrochemical detection. This is challenges we still have to work on. In fact, speaking of collaboration, we're now looking at microneedles because the interstitial fluids is a lot cleaner than a lot of everything else. That's another aspect we're looking at. So we have a microneedle expert at UBC in terms of the engineering side. What else? Pharmacokinetic studies. We need to do a lot of that. This is being done by Professor Anil Maharaj from the Department of Pharmaceutical Sciences again, and he's working with the AZAR again on this. In terms of internal standards, when I say better internal standards, the problem is you need something that actually behaves like fentanyl. We need something that electrodeposits the same way to have the best kind of standards. So if you look at fentanyl structure, one problem with maintaining the fentanyl structure is we have to make sure we're not going to make it super toxic. We don't want to just have yet another toxic thing as part of our container. So what we're doing is we're looking at different, I apologize, do you guys know what pharmacophores are? I know Professor Page knows the pharmacophore. These are the active components of the molecule. So if you look at all fentanyl derivatives, again, maybe sorry for those on Zoom. This will be over in a second. There we go. If you look at all the derivatives, you can kind of see they all have a similar pharmacophore, which is you've got the pyridine ring with the amino group in the pair position. Every single one of them does. You've got the amide over there. It's an alkyl. Some of them also have other derivatizations on this chain as well, but usually it's a similar piece. They all have the arene on the nitrogen. That's another pharmacophore. And they all have the 2-carbon fragment with some aryl group, whether it's alpha is a little different, but the same kind of gist. You've got the aryl group. If you start to modify any one of those core groups, you can decrease the activity significantly. So for example, norfentanyl is not very active because you're missing this pharmacophore piece. Okay, that's the gist of the problem. Now I apologize on Zoom. I'm going to zip forward to where I was. So what we're doing in internal standards is we're basically looking at removing pharmacophores, and I don't want to poison my students who are postdocs working on this particular project. At the same time, we need to make sure it has similar aspects. So what we're looking at is, again, we're derivatizing these sites. This is work that's done by Dr. Corey Bozzato. He's a postdoc. He's actually also working with another collaboration project we've got. Is working on computer policies company. The gist here is, again, we're modifying some of these so they're not as toxic. But even with that, we have naloxone kits in lab just in case something goes wrong. We tend to work under lethal doses, so what we anticipate these things to be. But the fun thing about doing this is we can also see what's going on. A peak at 0, I have to say, chemically is very weird. It doesn't make sense chemically, but why there is that peak at 0. So Dan and I are trying to figure out what's going on, and we can probe that by looking at derivatives of this. And we actually have I think we have a pretty good sense of what that peak is. Now, development of a prototype, as you can imagine, none of us have the experience to build it, right? This is out of our skill set. But Professor Bozzato with the analytical has a little bit more knowledge than I can barely run my laptop. So we need someone who can actually do this, right? So this was actually turned into a capstone engineering project. All right. So undergrads, we worked with the UBC and UBC engineering. In fact, 4 outstanding undergrads worked on this particular project. And we basically gave them the parameters of our device, what we needed. And they built a first generation opioid detector, right? And I know it looks a little complicated and will include you, but actually everything here is actually pretty cheap. Laptop is not that expensive. The syringe pump, all you really need is a screw drive syringe pump. There's nothing to that aspect. Most expensive piece, ironically, is the HPLC injector board. This thing's actually not that inexpensive or not that cheap. The flow cell, that's actually really cheap. In terms of separation column, we're just using little water separation columns. They're about $5 each. They're really cheap. The electrochemical piece itself, these are actually the screen printed electrodes we're using in this case as well. These are about $1 each. All right, the core technology, the potentiostat is $200. It's cheap. There's nothing cheaper. Since then, we've actually optimized this up a little bit more. We've improved the syringe pump. We've improved the flow cell design. The leaking was, of course, an issue. We now need this to replace that piece nice and easily. We've done that. We've figured out the electrodes. Some of the electrodes are more stable than others. Some of these screen printed ones are better manufactured than others. We had to work on that. Okay, and that's kind of the end of this particular project. This is where we're at. We've actually got a pretty good device in hand. It is ready now actually to go to, we're going to try to go to the safe injection sites to actually test real samples and do cross-validation with the existing methods to see how it holds up. So in terms of powdered samples, we're there. Fluids is still something, bodily fluids is something we're working on still. We're not quite there. Okay, so what will lead to game changing? And this is my little soapbox for what it's worth. What leads to game changing solutions and where do we need to go? So think of it as a lesson learned in everything I did wrong. Clinically driven research, I think, is going to be critical. All right. A lot of research groups, especially in, let's say, chemistry, physics, they tend to be so siloed that they kind of miss the point of what you're aiming at. Okay? Like we develop an interesting solution in search of a problem. And then we claim it solves something when it really doesn't help. I found actually just asking a clinician, what do you need? What's useful? What do you want to go for? And then that's where we can shine. We love to problem solve, and we can do all kinds of fun problem solving from there. But the clinicians also have to go to the fundamental scientists, because what clinicians, unfortunately, I think are limited to is they're limited to classic instruments. You buy an instrument, that's all you use. That's all you're familiar with. That's all you know about. Where analytical chemists can really help. They can tell you all different types of things to be using, new technology, and if it's on the market, this technology has been around for 20 years. We can actually step that up and do more useful and impactful things. This is where also broad engagement, why I like the Matrix N cluster and why we're working on other clusters as well. We need to get this engagement just so people know what the options are out there, what the current technologies are. And then I personally love collaboration, even in my core science. This is all actually ironically tangential to what I typically do, but even in my core projects, I think some of the best chemistry, some of the best science, some of the best medical advances all come out of combination. Last thing I'm going to do is just pitch an opioid research cluster. So Dr. Page and I started a different opioid research cluster, and we call it the ATOR. And its beautiful graphic is, of course, not me. This is all done by Dr. Page. I have no graphic design skills, so that's advantage on the team. The basic focus of this compared to Matrix N is we're really working on pharmaceuticals and detection. That's really limited. That's really the focus of our cluster. But fundamentally, it's still clinician-led. So we're basically Dr. Azar, Martha, they are helping drive in terms of what is useful, what can actually make an impact, what do they need to help address the issues they're facing. Not just what can they do with existing technology, but what if this technology existed? What could I do? And then in terms of the team, we have a broad team of both chemistry and pharmaceutical science. I'm just going to start with the chemistry. Professor Laurel Schaefer, she and I actually have a very similar expertise, except she's a little bit more on the catalysis side. She's a little bit more inorganic, but she does a lot of alkaloid chemistry as well, and interesting, really fancy and clever ways to make carbon-nitrogen bonds. And she actually has an opioid projects as well. Professor Jason Pine, I've talked to him a little bit about him before. He actually does automation. And in fact, another project he has on terms of an opioid project is he's teamed up with a cheap mass spec. They have a robot that basically samples things for you. So rather than people needing to handle these samples, you got a robot that just takes care of everything. In fact, the police are quite interested in that right now, just for seeing what's on the market, trends, and they have all the analytics in terms of the output of this. Dr. Reed, she does computational organic chemistry. So if you need any kind of docking models, and she does competition's fantastic. He's kind of a rising star in the field. Djavad talked about him extensively for electrochemical analytical side. Russ Alger, he's a professor also in analytical, but he does a lot of diagnostics. So he works on photodiagnostics. So basically, bench to bedside, you can take your cell phone and do analytics just with the cell phone technologies. In terms of pharmaceutical science, Peter, our lead and drug delivery is his primary expertise, although he has some other synthesis side as well. Neil Maharaj, he does the pharmacology and pharmacokinetics, and he's an expert in that side of things. Then we have pharmacists on the team. Again, because we have the issue in British Columbia with the broad diversity of work we have, we also have the pharmaceutical care with BC First Nations as well. We have that side as well, which we need to incorporate in a lot of our solutions. And with that, I just like to thank the people who basically helped drive each one of these projects with me. So Anthony Phillips, of course, has been, without him, Govadine would not exist. That project would not be there. So he is fundamental for that. Jeet Arlene, Brent Page for a lot of peptide projects in that direction side of things. And then for the detection side, Dan Bazzato, Azar, and a lot of other people, as you can see, are starting to collaborate with that aspect of things as well. And then I would be remiss if I didn't mention my own group. These are my current research group funding sources. And you've seen, I kind of talked about them as I went through things as well. But I'd just like to thank you for your time, and I'm happy to answer any questions.